Okay, is it recording now? It is good. Okay. So as we gather today to celebrate Lucy's memory through this seminar series that began 1 year after her passing, and it runs twice annually, let us reflect on her impact on her science and the generations that she continues to inspire. So we have had two in the past 1 year. And in September 2023, we had Dr. Amanda Simcox, and in April 2024, we have Dr. Stephanie Moore. So it is therefore my pleasure today to introduce today's speaker, Dr. Shinya Yamamoto, an associate professor at Baylor College of Medicine and an investigator at the Jan and Dan Duncan Neurological Research Institute at Texas Children's Hospital. With a background that spans both veterinary medicine and genetics, he holds a Doctor of Veterinary Medicine and a PhD in developmental biology. He brings a unique interdisciplinary approach to studying genetic diseases. One of his major contributions has been the development of a human cDNA library that we are going to hear a lot about today. And by humanizing fly genes, especially replacing fly genes with the human equivalents, we can functionally study human genetic variants in a relatively simple model organism like flies. This approach has facilitated the diagnosis and understanding of rare human diseases. And through his work with the NIH Undiagnosed Diseases Network, the UDN, where his team has identified over 30 new disease-causing genes. His lab, funded by the NIH and the Chan Zuckerberg Initiative, uses Drosophila to unravel the functional consequences of genetic mutations that cause neurological and developmental disorders, thus providing insights that cannot be easily obtained from human studies alone. In addition to his research, Shinya is a dedicated mentor, shaping the future of genetics through his teaching and training programs. So lastly, a few guidelines for this recorded seminar. Please type your comments or questions in the chat box, or raise your hands if you have questions during the seminar to ask. We will either then have you ask it directly or relay your questions to Shinya. And this seminar will be recorded, and the recording will be made available on our website. Please join me in welcoming Dr. Shinya Yamamoto, who will speak today on facilitating rare disease diagnosis through functional studies using Drosophila. And Shinya, please. I'm going to stop.
Thank you. Yes, and let me share my screen, and let's see if the screen, do you see the full screen here?
Excellent, yes.
Okay, great. Well, thank you, Arthur, and everybody from DGRC to invite me to this wonderful lecture series honoring Dr. Lucy Cherbas. My name is Shinya Yamamoto. I'm at Baylor College of Medicine, also affiliated with Texas Children's Hospital. I have also a role in the Undiagnosed Disease Network, which I'm going to be talking today. And there's also an international version of that in UDNI, Undiagnosed Disease Network International, that I also play a role in. So as Arthur mentioned, we are here to honor Dr. Lucy Cherbas, whose, I think, the seminal contribution to the field has been primarily in the field of tissue culture using Drosophila. For example, I'm just highlighting one of her work here. There was a whole issue that was dedicated for the Modern Code project right here. You can see the beautiful picture of flies as well as C. elegans here. And one of the highlighted articles here is really to characterize 25 Drosophila cell lines that she has collected over the years and also distributed to the community through transcriptomic analysis, which led to a really decoding of the genome, which was what the modern code was there. She has also been always interested in adopting new technology. For example, this paper in genetics was actually integrating at the time, the latest 5C31 technology to really manipulate the Drosophila cell lines in a very specific manner. As we heard the two previous lectures, the first one, the inaugural one given by Dr. Amanda Simcox, have really highlighted also the importance and value of these type of resources for the field. And then in the spring lecture, Dr. Stephanie Moore elaborated on that and also introduced how these cells are actually being used for high throughput screens and also being a resource for the entire community. If you have not seen this lecture, these are also available on the DGRC website. I put the QR codes there. And throughout my presentation, I think now the younger generation are used to these QR codes. I'm just going to be putting them throughout my talk, and feel free to take screenshots or anything. So the theme of this, I think, Lucy Cherbas seminar series was initially started as let's highlight the work of, especially in the field of cell tissue culture and cell culture in Drosophila. And although I have actually used S2 cell, for example, part of my thesis project that I published on notch signaling, for example, my primarily use of Drosophila was always in using it as an in vivo tool. And I think I'm actually invited here, and Arthur alluded, I have been, but sharing some of the ideas that Dr. Cherbas had, which is the importance of collecting resources, and generating new ones, and also widely disseminating it throughout the community so that we actually give tools and resources for people that wish to generate, actually perform creative experiments. And my specific contribution, I shouldn't say my because I think it's our. I have been collaborating with Dr. Hugo Ballin here at Baylor and Dr. Sue Selnicker at Berkeley, Lawrence Berkeley National Laboratory, to generate a large collection of clones and also transgenic lines that allow the spatial and temporal regulated expression of human proteins. If you go to the DGRC website, and here you can just click on this catalogs of clones, you can find this link right here called Human Orf. So if you click that, it would actually allow you to download an Excel file. And you can see right now, I think if I looked yesterday, there's 5,317 unique clones that are available from DGRC today, that are containing these human cDNA in a PGW HA@B plasmid. So they're ready to be generated, injected, or you can actually take this, do some site-directed mutagenesis, make sure that you don't produce any second site errors, and there's ways of doing that. But you can actually start introducing any type of variants you want in these construct and either use it in the cell culture setting or make transgenic flies out of it. I think the highlight of our collection is the price. $52 per human cDNA clone is a bargain compared to what the other commercial entities offer. And there's also about 2,000 additional clones that have been generated through Sue's lab through high throughput screening cloning method using robotics. There is a total of 7,500 human cDNA clones that would be available to the fly community for a really decent price. This, I think, if you kind of look at the human genome, there's about 20,000 human encoding genes. People say there's about 60% conservation of human genes in flies, and I think we've been prioritizing genes that are more highly conserved. So this actually tells you that we are covering the majority of human genes in this collection. We can talk about like different splicing isoforms and stuff like that if you have any questions, but that's our collection. So I don't know if I can. Sorry, my Outlook is giving me a reminder. Also, we took many of these clones that we generated as plasmid and collaborated with Dr. Toshi Takano at Kyoto Institute of Technology. He's the director of the Kyoto Stock Center, to try to generate as many UAS fly cDNA transgenic lines as available so that people actually have off-the-shelf reference, which we call it as AKA wild type human proteins that people can use for overexpression or rescue-based experiments. And if you count the numbers together, all the lines that you can find in BDSC, the Bloomington Drosophila Stock Center, and which is also in Bloomington, as well as in Kyoto Stock Center, you can actually find probably many of the genes that you're interested in already available as a transgenic construct. We still need to publish this case, but all the lines are made available prior to publication. So I think this reagent, I want to actually, use this opportunity to explain why we generated this resource and how do we decide to do this for the community. And in order to do that, I do want to introduce you. Some of you might not be familiar with the concept of rare and undiagnosed diseases that many individuals in the world are struggling with, and how this puts burdens on patients or families, and as well as the entire society, and why is it important to study these conditions. And then I'll actually switch to explain my personal story, why I got into this rare and undiagnosed disease research, and show that how experiments and resources in Drosophila can be used to discover and study new human disease genes. And this is actually not rocket science. A lot of you can do what we're doing. I think the key is you need to find good collaborators and also have a good knowledge of not only Drosophila genetics, but human genetics and biology. Also, starting from this, although we started in, primarily been focusing on these rare and undiagnosed diseases, we'll show you a couple of examples of how similar approaches in flies can be used to study human genes and variants that are linked to more common conditions, such as autism. And I'll also touch upon our recent collaboration with Ethan Beer's lab on generating a collection to understand the pathogenesis of COVID-19, which sounds like it's something that people wouldn't work on flies. And finally, I want to actually end up this talk by discussing how you actually, as a Drosophila researcher that's working on maybe some really biological fundamental question, can also be involved, dedicate part of your time to this collaborative research with clinicians, and also how it's important to leverage the resources that are available from these public stock center, including DGRC and Bloomington Stock Center. So just a little bit about rare diseases. So the word rare diseases kind of have a legal meaning. And in the United States, there's a law that says that determines what is a rare disease. So in the United States, a disease is defined as a rare disease or an orphan disease when it affects fewer than 200,000 people. So if you think about the population of the United States right now, it's just about 300 million people. One disease affects 1,500 or less. So, and sometimes it's actually 1 in 10,000, 1 in 100,000. It could be one disease that only affects 1 or 2 individuals in the world. So, if you think about like how many people you are connected in LinkedIn or Facebook or TikTok or whatever, I think that means that there's very few people that have like friends more than 1,500. So for an individual disease, it's very rare to have actually someone that you know. However, there are approximately 7,000. I think the number is getting close to 8,000 because there's new discovery happening on a daily basis. There are many, many different types of rare diseases. So if you add all this, numbers together with these 8,000 condition, which all of them are rare, actually the math says that about 1 in 10 Americans are affected with some kind of rare disease. Different countries have different definitions for them. For example, the European Union has a definition of a rare disease as less than 1 in 2,000 individual. What we're talking about in the same ballpark. The problem of these rare diseases, many of these rare diseases are very severe. Approximately half of them are affected or show symptoms as children. And unfortunately, 30% of these rare disease children will not live to see their 5th birthday, which is very sad. Because individual diseases have very few individual, because many pharmaceuticals company used to think that these are not profitable, so nobody would have thought of developing drugs for a disease that only affects, for example, 10 people in the United States. So that's why even currently 95% of rare diseases have no FDA approved drug treatment, and only care that can be done is really treating their symptoms and not really getting to the base of their condition, which is actually changing because there's a lot of interest in the biotech domain about targeting rare diseases. And there's more newer technologies like antisense oligonucleotides, gene therapy, cell therapy, and all these things are coming. So this number would be coming higher as well. However, it's still that because that you need kind of personalized therapy for these conditions, many of these people do not have really a good way to deal with their conditions. And also, there are many different kinds of rare diseases. There's some diseases that are purely environmental, or there could be very rare infectious diseases, but it's thought that about 80% of these rare diseases have a monogenic origin or a strong genetic component that is cause of their symptoms. And these numbers are coming from statistics that are gathered by patient advocacy groups such as NORD and EveryLife. So obviously, if you have somebody that and you yourself or somebody in your family that has a rare disease, that's a big problem. And the first problem that they actually face is called a diagnostic odyssey. So because their condition is so rare, and maybe usually physicians that are practicing have never seen these individual for specific conditions, they usually take a long time to first of all get an accurate diagnosis. So if you kind of interview individuals who have received a rare disease diagnosis and ask how many years has it took you to get a diagnosis, the average is about 6 years, and they said that they went to 17 different doctors or hospitals to receive a diagnosis. Obviously, if they have received a diagnosis in their first visit, this would have saved a lot lot of money, and one calculation is about $220,000 could have been avoided if people didn't have to go through this diagnostic odyssey. And this is actually statistics from people that did end up getting a diagnosis. And the problem is that even if you go through this diagnostic odyssey, many end up not getting an idea of what the cause is. And so 50% of these individuals still remain undiagnosed after these extensive clinical workup. And if you think about 30% of the children that are affected do not see their fifth birthday, which is actually beyond lower than this average number for diagnosis, you can see that many families lose a child without knowing what the cause of their death was, for example. And that also is a big grief for the family, but also leads to their next question. Okay, what is the chance of my second child, if that was the first child died, having a chance of having the similar condition? Well, if it was a recessive condition, yes, you have a 25% chance of getting a child with that same condition. If it was X-linked, it would be 1 in 2 boys would have that condition. Whereas if it was a de novo, a single isolated mutation event that happened in the germline, the chances of that recurrent mutation is very, very low unless you have like a clonal event that is in the gonad. So it's very important to know, even if the patient has passed away, understand the condition for genetic counseling purposes. The other thing is that, well, even if it's not you that actually are going, you or your family that's going through this diagnostic odyssey, this is actually impacting the society. So every time, obviously, these families need to go to, doctor's visit, they'll get tests, and they need to go repeat these tests and over again. They also need to, take days off of school and work. So there's actually a huge economic burden on society, both medical and non-medical costs. So one estimate was that it's $1 trillion are being spent on these patients with rare and undiagnosed diseases, including not only their treatment, but also to their effort to get a diagnosis and management. Also, one scientific reason for why we should pay attention to rare diseases is often that these rare diseases shed light on the mechanism of more common diseases. So you can think about, because these are really, a lot of these conditions are monogenic, there's a clear genetic genotype phenotype correlation that you can actually determine. And for diseases that actually are polygenic, it's very difficult to figure out where the problem is. But these studying these individual genes that are associated with rare diseases often provides a clear-cut mechanism as what's happening at the pathogenesis level, at the cellular and molecular level, which you can elaborate to understand a related condition. So more recent studies, for example, suggest that lysosomal storage disease is actually a very devastating neurodevelopmental or a multisystemic disorder. And then this is a recessive condition primarily, but the parents of these kids who have lysosomal diseases are at higher risk of Parkinson's disease. And it looks like lysosomal deficiency and also mitochondrial deficiency are underlying cause of Parkinson's disease. And similar thing has been found for association with Lee syndrome associated genes in GWAS studies that are hit in Alzheimer's disease. So this actually also opens up a lot of opportunity for biologists to start cracking these more difficult to study common disease conditions where both genetic and environmental factors converge to get you a certain phenotype. Now is a time to actually do these type of research, because one key thing that have been happening in the human genetics field is the revolution of the genome sequencing technology. So if you go back, I don't know, 30 years, the Human Genome Project started in 1990, and they kind of completed the human genome. This was kind of the first draft that was completed. There was also this telomere sequencing that was completed more recently, but this initial draft of the human genome, where they actually sequenced, I think, 95 or 99% of the human genome, it costed $2.7 billion. So it was obviously, if you use the technology at the time, it's going to be difficult to actually sequence individual genomes. Craig Venter sequenced his own genome through his institute, and this was using the previous technology Sanger sequence, which is still used, but not high throughput. It costed him to $150 million to sequence his genome and let it out in 2007. And also there was also a revolution in the field where actually they went away from Sanger sequence and tried to look at other technology that can do sequencing. And everybody uses Illumina sequencing nowadays, but one of the pioneering companies, 454, had this technique called pyrosequencing, sequenced Jim Watson's personal genome for $1.5 million. So as you can see that the cost of these, following Moore's law, the cost of sequencing has been dramatically decreasing over time. And how much does it cost right now? It's like this was a number I found from 2022. It costs about $1,000. So either do a 30x coverage of whole genome sequence or 100x coverage of whole exome sequence. This is the kind of the quality or the metric that you need to actually identify these heterozygous variants in the human genome. $1,000 is actually a realistic value that people can pay, or insurance or even Medicare can cover in some cases. So nowadays, a lot of people that have undiagnosed conditions, instead of doing gene panel screenings, they sometimes go straight to whole exome sequencing. If it's negative, they might even go to whole genome sequencing. And this has been becoming, especially in the clinical genetics and maybe in pediatric especially, a routine clinical agenda. And this is actually thought to go down even more dramatically. So there was this article a couple of years ago, and there's this stealth company that was in the biotech that is coming in from a different way of sequencing, and they claim that they can actually sequence the human genome, personal genome, for $100. And Baylor has been working together with them, and it looks like it is something that is really happening. My department chair, Brendan Lee, says that at one point, getting a coffee in Manhattan, New York City is going to be much more expensive than getting your genome sequenced. So that's where we're actually headed. I think sequencing somebody's genome is going to be very, very cheap, and it could be done as a standard clinical practice. Even there are countries that's thinking about doing it as a newborn screen. So that's all great. We can now sequence everybody's genome that's on this call right now. But there's problems. So every time a new technology is introduced, there's always problems. And the problem of the whole genome technology, so sequencing technology, is that you see too much. If you are dealing with gene panels and you have to look at variants in maybe 30 genes that are known to have associated with your condition, you can do that manually. But when you actually look at individuals' genomes, there's a lot of things in there. And also, there's also a problem with lack of knowledge about gene function. For example, we don't still know what most human genes do in vivo. So why did I say that? Well, there's a bunch of papers if you go on PubMed, but if you look at really a database like OMIM, this is the Online Mendelian Inheritance of Men. This is a central database for genetic conditions, both disease as well as hereditary traits in humans. And there has been about 5,000, a little less than 5,000. I looked it up yesterday. 5,000 genes, human genes, that are associated with some kind of a visible or kind of a behavior phenotype. This is not including studies where, like GWAS, where we don't necessarily know what genes is causing. You have a SNP that's associated with a phenotype, but you can't really attribute it to a specific gene. So if you include those, there could be more. But these are kind of like clear, human variants that are, germline variants that are associated with relatively strong phenotypes. And out of about 20,000 human protein coding genes, that's actually present. So if you think about it, we can say, well, maybe for 25% of genes, we know something about the human genes in vivo. So regarding the remaining 70% of the gene, human geneticists even use the word genes of unknown significance, so GUS. Some people actually, I saw the people call it the ignorome of the human genome, because people kind of tend to ignore it when they see it because they can't really make sense out of it. On top of that, we know as fly geneticists here, most genes are pleiotropic. There's not going to be like in not the old days, like one enzyme, one gene hypothesis. Most genes are pleiotropic. One gene can have different roles in different tissues, or even an enzyme could have a moonlighting function, maybe as a chaperone, maybe as a secondary protein for forming a complex, and they can be altered in different ways. So obviously, if everyone knows Miller's different morphs, for example, a gene can be mutated to become an amorph, complete loss of function, hypomorph, a partial loss of function, antimorph, like a dominant negative, hypermorph, a gain of function, too much activity or too much protein, or even neomorph, having a new function. And if you think about these different morphs in flies causes different phenotypes, if that happens in humans, different variants in humans could cause different phenotypes. And when we talk about phenotypes in human, those are typically diseases. So when you look at these 4,929 genes that are associated with a human disease, I can find today 3,453 genes that are associated with one disease, whereas 1,476 genes that are associated with 5 or more. And some diseases are associated with maybe some, like 5 or sometimes up to 10 different diseases, depending on the strength of the alleles and whether there's a gain of function or loss of function. You can come up with different disease type. And these are very distinction of these different variants are very important. So if you say that you have a patient with a variant in gene X, if it's a gain of function, if the variant is a gain of function, your treatment, your therapeutic treatment should be to downregulate that gene, right? Versus if the patient actually has a loss of function, then you need to augment that. So doing that, for example, selecting whether you want to go for an ASO, antisense nucleotide therapy, or gene therapy, it really depends on the type of variant that you have. And on top of that, everybody is unique and you have some kind of a variant that's never seen in any other individual. We cover so many rare variants and their significance are almost unknown. So if you kind of compare your genome with your parents' genome, probably you'll have 10, about 10, maybe what we call de novo variants, maybe ranging from 0 to 10. And maybe 1 or 2 of them are encoding regions of the gene. So even if you look at a healthy individual, what we call healthy individual, you often find a de novo missense variant or sometimes truncation variants in a gene. And if that gene has never been linked to human disease, it's very difficult to interpret that. So what you're trying to do when you're sequencing somebody with a variant and the whole genome sequencing and data in front of you and all the phenotypic information, it's really a daunting task to figure out what are the phenotype that which variant in your genome can explain which phenotype. And sometimes, and also this happens as well, sometimes patients have 2 diseases that are blended. And maybe some of the phenotypes is probably gain of function variants in gene A versus another phenotype that's unrelated is coming from loss of function in gene B, but because that they're blended, it's very difficult for the physician to say that whether that's one or the other or both genes are contributing. And there's always other things like genetic interaction, suppressors, different expressivity, penetrance, and everything that we can kind of go through, talk about genetics in Drosophila happens in humans as well. The reason that I kind of got into, and this was very serendipitous, I was doing my PhD in Dr. Hugo Bellin's lab here at Baylor College of Medicine. And third year of my grad school, I was kind of in a slump. It's like nothing really worked. A lot of the projects that I started when I joined his lab in second year actually went down the drain. And I was really kind of starting to get depressed about like, what am I doing here? And it's like, what can I do? And at the time in Hugo's lab, people were doing 4 genetic screens using chemical mutagens, EMS, doing clonal mutagenesis screens. So mutagenize an FRT chromosome, make clones in the eye or the wing, and then the thorax, look for morphological phenotype or electrophysiological phenotype. And then once you find some interesting gene, we basically try to clone it and figure out what the gene does. At that point, I started saying, well, maybe, it's time for me to do a new screen and let's do something that people haven't done before. So I decided to do a random mutagenesis screen on the fly X chromosome, which was actually for a chemical mutagenesis screen for lethal mutations. People didn't want to touch it because it was a pain to try to clone the mutation. Even if you have a very interesting variant, it was very difficult to clone it. But we came up with a strategy to do a clonal screen. It was an F3 screen, and we also were able to leverage the newest technology of whole genome sequencing, the capacity that we had at Baylor, together with back transgenic constructs. And also Bloomington also generated a bunch of new duplication lines that allowed us to really characterize essential genes on the X chromosome and look at their clonal phenotype in multiple tissues, including the nervous system. We actually used the eye and did electroretinogram on these mutant clones in an otherwise heterozygous animal, so that you actually have the animals living. However, you could get patches of mutant cells that are mutant. And we can look for defects in not only in neurodevelopment and synaptic transmission, but also by doing this in an age-dependent manner, we were able to look for new genes that are associated with neurodegeneration. So this was an interesting screen. We identified 165 genes on the X chromosome that was essential and had some interesting nervous system phenotypes. I mean, when I was trying to wrap up this screen, we were kind of intrigued by the fact that about 93% of the genes that were identified from this random mutagenesis screen were evolutionarily conserved. And at the time, 2012 or something, 30% of the genes were associated with a genetic disorder. So we thought, hey, well, this is cool. Well, we can actually have this as a collection of mutants that are can be used to study these evolutionary conserved genes. And for the 30% of the genes, you can actually study the mechanism or the phenotype and then try to make links between the genetic diseases that it was associated with. And all these collection is actually can be ordered. Representative alleles of this screen can be ordered from Bloomington. Also, all the lines that are still some, including some unmapped lines, there's still about 1,200 lines, and maybe half of them are unmapped that are actually being generously kept by the Kyoto Stock Center. And we're trying to sequence the remaining to kind of make them available to the community. But what we decided to do was actually, well, let's say that 30% of these genes that we got from this screen were associated with the human disease. What about the remaining 60% right here? They're conserved in humans. Nobody said that they were associated with human disease. Is it that they're not associated with human disease, or still geneticists have not discovered them as really human disease causing? And this was actually coming from the same question. Like if you sequence an individual's genome, you see so many variants, and you can maybe they could come up with the top 5 candidates in that genome, but they're kind of stuck there. So what we did, and we collaborate with Dr. Jim Lipsky and Richard Gibbs, together with also Rui Chen, who's been doing the fly sequence for us. We were trying to actually reach out to them and say, hey, we heard that you're sequencing a bunch of patients with these rare diseases and have some candidates in certain cases. Can we actually cross-validate our list? And can we look to see if you guys have patients with variants in the genes that we're interested in? This actually led to a really interesting the series of discoveries where we identified patients who have what we would call deleterious variants or mutations in the gene of interest that have phenotypes that sometimes are very similar to what we see in flies. For example, a gene called ANKL2, we identified as a novel microcephaly causing genes. A gene called OGDHL and nardolysin was associated with the neurodegenerative phenotype in flies. So this was really good and showed the proof of principle that you can actually use flies as a new disease discovery tools. And this dialogue between the fly geneticists and clinical geneticists, if you can go back and forth, this is one very productive way to leverage the knowledge that we have in the fly field, because many of these human geneticists cannot even read a fly genetics paper. So this was cool, and we were very excited, and so was the NIH. So the NIH was at the time launching this national network called the Undiagnosed Disease Network. This originated in the Bethesda as part of the NHGRI. And Dr. Bill Gall started this as an intramural program called the Undiagnosed Disease Program. And the idea was that they wanted to accept patients, individuals who have been undergoing this diagnostic odyssey, bring them to the clinical hospital at the NIH, let them have a head-to-toe phenotyping to kind of understand the clinical condition, get them also the latest biochemical tests and whatever you can do to phenotype these patients extensively, and also genotype them initially with whole exome sequencing. But as the technologies emerge, we're now doing whole genome sequencing. Sometimes affiliated also, we're doing accompanied with RNA sequencing type experiments. For this initiative, they were actually starting to enroll patients. And today I looked at the database, there's about 7,000 individuals who has applied. Their data becomes integrated at the coordination center, sent to different clinical sites across the United States. And actually, Indiana University at Indianapolis has actually been recently selected as one of the clinical sites. And they're basically assigned to one of these clinical sites. And there has been about 3,000 individuals that have been accepted. So there's about 40% acceptance rate. And I can tell you why some people are not accepted into this study if there's any questions. But after these patients are accepted, they'll actually, again, undergo similar to what was happening at the NIH, head-to-toe evaluation of these individuals, as well as whole genome sequencing. There's also a metabolomic score at Mayo Clinic that can do targeted or non-targeted metabolomics, especially for patients where we think that there has a suspected metabolic disease conditions. But then even with all these technology, you may be able to identify the cause of maybe about 20% of these individuals. And again, that 20% is big because these people that have been accepted have really exhausted all their clinical option to get a diagnosis. So even, giving an answer to one family is actually a big thing, especially for that family. But the problem is, okay, once you do that, you're stuck with all these, candidates that you get from the whole exome sequencing. But sometimes, and oftentimes, actually, clinical sites can identify something that's very suspicious. Oh, this is a variant in a gene that has never been associated with a human disease, but the variant looks like this deleterious. If you look at the population genomic data, there's not many people carrying variants in that gene, so there's a selective pressure. How can we study that? Or it could be a situation where, the gene has been linked to human disease. It's been linked to a recessive condition, but now we're seeing a patient with a de novo heterozygous variant, and the phenotypes could be somewhat related. Maybe then the variant is a dominant negative protein, or the variant is very different. The phenotypes of the patient is very different. Oh, maybe it could be a gain of function variant that has like very different phenotypes from a loss of function. What can you do from here? So the NIH was looking for solution to study this functional consequence of genetic variants, and they heard about our story as well as some emerging stuff in the zebrafish field. They decided to establish a model organism screening center that is going to be that works with the clinicians at the Undiagnosed Disease Network. And this was an open RFA that was out, and together with a zebrafish group in Oregon, Hugo Bell and myself and Mike Wangler got this grant to start the first inaugural model organism screening center for the UDN. Later on, 3 years down the road, they found that this actually approach was successful. They want to expand it, so they added one more MOSC site that uses C. elegans as well as brings in additional capacity and different expertise in zebrafish. So now we're using 3 model systems, C. elegans, Drosophila, and zebrafish, to functionally study these variants that are associated with rare human diseases and likely to be candidate genes and variants. One of the things that we did develop over time is that we needed actually a way to really have an effective communication between clinicians and our scientists here. And although I do have a veterinary degree, and Hugo Bellin also has a veterinary degree, Mike Wangler is an MD himself, we are able to kind of talk the language of medical language. However, even with that, I think it's very important that also we are able to kind of communicate what is known about these homologs in this gene. First of all, is there what is known about the human gene that the clinician submits us as a good candidate? We need to do our homework to make sure that we also agree with them. What are the orthologous genes in the different model organism? Are there any types of do these model systems actually do have orthologous genes, or sometimes the gene is absent in C. elegans and Drosophila, so therefore it needs to be tackled in zebrafish? And also, the variants that are nominated, mostly we primarily work on coding variants, and actually that's for a technical reason. Again, I'll be happy to elaborate if you have any questions, but still we have a lot of missense variants that needs functional annotation. So these missense variants are very difficult to predict whether they're damaging or not. So we have these kind of questions. Initially, in order to actually answer all these questions, we were opening up a lot of different browsers. For example, there's a very useful tool called DIOP that actually Claire Hu, I saw it in the audience, has been developing at Harvard, which actually selects the best ortholog candidates in different species. But that actually is kind of a gateway for us to kind of connect the human gene-based information. What is the gene known in disease databases? What is the gene known in control databases? And also together with model organism-based knowledge. I'm very familiar with navigating Flybase, but when I go to like ZFin as well as Wormbase or even MGI, I'm actually kind of lost to kind of try to get an information. But all these things are very important for us to make a decision which model organism is the best and what methodology would be the best to answer these questions. But so I'll show you a little bit of a demo of this tool called Marvel that we developed in collaboration with Zandon Liu's lab here at Baylor. But this is kind of a one-stop shop where you can put in the gene of interest and also the variant of interest and gather all this information. If you don't know the human gene you can actually start your search with the fly gene and then try to find the human gene and then try to get the information. I'll show you in the next slide. But once you actually pass that, so far about 407 genes have been submitted to us, covering about 450 cases. And we have prioritized them, and we accept about 50% of the cases based on the availability, as well as also we assign them to different model system as well. So we prioritize so the majority of the cases are coming to the Drosophila core. You can see 118 cases have been assigned, 51 cases in zebrafish core, fewer number from C. elegans core. That's why the reason being that they've kind of joined the network a little bit later, but also their technology allows them to only study variants that are conserved in C. elegans or not. And actually, Brian, thank you so much. There's actually also a link in the chat that Brian Calvi actually put in. He's actually one of the leaders of the Flybase. There's another very useful resource for the community called Alliance of Genome Resource, AGR Alliance. where they try to also integrate multiple database like C. elegans, Drosophila zebrafish, MGI, so that you can actually have one user interface and gather all this information. And we think that the Marvel 2 is very complementary, especially because we try to curate in a lot of human information as well. And I'll show you here. So this is Marvel. Make sure you put in 2 R's unless you're trying to watch Spider-Man on Google. Through Google, but here's like, so you can actually, this is a very simple user interface. You can enter as human gene or human protein based nomenclature. I mean, the difference is that you can actually put in, especially if you're looking at a specific mutation or variant, you can put in the nomenclature like alanine 153, phenylalanine, for example, or you can start the search by model organism gene. And if you here, this is quite a while ago, but I was trying to look for a gene called GLRA2, which I was interested in time, and try to do a search. Here, there's a brief description of what GLRA2 is from NCBI. At that time, this gene was never been associated with any disease, so we curate information from multiple disease associated with databases like OMIM, ClinVar, Geno2MP, Decipher. There's also information coming from control database. So there's large population genomic database in humans that you can actually see the allele frequency of variants. Here we are showing DIOPT data about different model organism ortholog candidates. Also where the gene is expressed in humans using GTEx, where the gene is expressed in model organism based on model organism database, again, borrowing data from AGR, phenotypic information from individual model organism databases that are kind of assigned to large human phenotypic categories, whether they're drugs that are identified that targets these as ligands, and also show you the domain structure as well as a protein, multi-protein alignment. We also have integrated information from AlphaFold to kind of get you an idea of the structure of the protein. And again, for this talk, I don't have time to kind of go into really how to play with this, but I gave a talk several years ago on a GSA webinar series. So you can actually watch this in YouTube if you're interested, about maybe half an hour that goes through different categories. So once these cases are assigned to us, we basically work together with a lot of trainees, primarily postdocs and graduate students that are associated by a lot, but that are supported by a lot of technicians. And this is a collaborative effort between Mike Wangler's group, Oz Kanja's group, Hugo Ballin's group, and my group. And this is just a snapshot of one time, and there's always people coming in and out, and there's actually maybe more people that's associated. And just to clarify, our task at the functional study core is to really assess if the candidate variants that were identified in the patient have functional consequences using Drosophila. So what this means is that we're not always trying to generate a disease model. When we go to talk to clinicians saying that we work on model organism and they start questioning us, so is fly a good model for certain conditions? You know, I'm studying bone disease. You know, what can you do in a fly which doesn't have bones? Well, we can say, well, flies don't have bones, but tell me what your gene is. Oh, it's a TGF-beta or BMP protein. So BMP is like bone morphogenic protein. Yes. Well, we have the homolog of that called DPP in flies. And although flies don't have bones and cannot cause like maybe osteoporosis, we can actually tell you, if you tell me a variant in a gene that is in the pathway, we can see if the gene is conserved and whether that variant, a missense variant in the gene, is a gain of function or loss of function. So I kind of use the analogy that we're using flies as a Leving test tube to perform functional assay to kind of see whether the variant that you're interested differs, first of all, from the reference protein. And if it does, which direction does it differ? So the key of this is really the technology. And I need to really emphasize that there's 2 really big components of this, which is a resource that allows us to generate loss of function alleles that also have additional features, and also these UAS human cDNAs that we talked about. And one of the main technology that we use is to try to attempt to humanize a fly gene. So what do I mean by that? So conceptually, what we're trying to do is we're trying to create a loss of function of the fly gene, but then reintroducing the human protein, first the wild type protein, and see if you, first of all, does the fly loss of function have any kind of scoreable phenotype, regardless of whether it has anything to do with the patient phenotype. And if you see any phenotype like lethality or sterility or behavior defects or maybe morphological defects, we ask the question, can the human proteins rescue it? And we need to we want to rescue the human protein by reintroducing it in the same spatial and temporal pattern of the fly gene. So that's the concept. So how do we do that? Well, for genes, like a typical fly gene that has exons and introns, oftentimes you have large introns. We integrate a cassette called a T2A Gal4 cassette. And this technology was pioneered in Ben White's lab at the NIH, using also leveraging a mimic technology that was originally developed in Hugo Ballen's lab by Kuhn Venkin. So in this case, you can integrate an artificial exon called a T2A Gal4 cassette, which has a splice acceptor in front of it and then a pulley A tail behind it. So once this cassette is integrated, what happens is that the splicing machinery would think that this is a real exon, and then the RNA polymerase would drop up right here because they think it's the end of the protein, leading to generation of a truncated portion of the protein that also encodes this T2A Gal4. T2A is a ribosomal skipping peptide. So the ribosome, when this RNA is translated, you do make the N-terminal portion of the protein of interest. Most of the cases, like 95% of the cases, this is a strong loss of function. And you also generated a Gal4 that which is not physically separated from the original protein. So it can go into the nucleus and do whatever it wants. For genes that are small, we actually also can knock in a Gal4 using also, we call it the Kozak Gal4 system because we also put an artificial Kozak sequence to enhance the translation of Gal4. But basically what this allows you to do is to create a loss of function of the fly gene, but also generate a Gal4 that expresses in the same spatial and temporal pattern as a fly gene. So everyone here, Brandon Perrimon, 1993, everybody knows about the Gal4 UAS system. So if you bring in a UAS human cDNA in this situation, and make it either in the I would kind of try to avoid working on a homozygous background. So we typically do it over a deficiency or over a known as mutant allele to avoid for second site hits. But what we can do is in a loss of function background of a fly gene, you can reintroduce the human protein. And also, when you actually show that there is a beneficial effect, it doesn't have to be a complete rescue, but if the human rescues to a certain extent, you can then compare how the reference and the variant behaves. And one important thing is that the reference and the variant proteins, transgenic lines are put in the same docking site. So we are using 5C31 mediated integration. So a large collection, about 3,000, 4,000 fly genes are already tagged with these Kozak Gal4, T2A Gal4 systems, thanks to the efforts of Dr. Bellin, Kanja, Perrimon, and Spradling's lab. And also so Dr. Salniker and Hugo Bellen's lab and my lab together with Takano's lab have been generating a collection of these lines. So for a lot of cases, you already have kind of a pair of T2A Gal4 with the reference human cDNA. So you can actually try to look at these databases to see if you can start trying to humanize your gene of interest. Does it work? Well, our experience, roughly, I think 60% of the fly genes that we have tested have been successfully humanized. And obviously, other ways of humanization exist. For example, you can knock down the fly genes using RNAi and then reintroducing the human cDNA with the same Gal4, for example, or you can humanize the genomic rescue construct or do a knock-in. Well, this is all good, but what about the 40% of the genes that doesn't work? Well, it's I'll talk maybe afterwards, but after giving you some examples. So this is one study where we did this type of humanization very early during the UDN. This is one of the first patient paper that we published in 2017. Tuan Chow has been continuing to work on this gene called EBF3, but the clinical sites actually at the NIH identified a patient with a de novo missense variant in this gene called EBF3. The patient had developmental delay, intellectual disability, some MRI findings in the brain, primarily a neurological phenotype. And then we started working on the case to characterize what this R163XQ variant does. One thing that's important in human genetics is that we try to identify other patients with similar genotypes and phenotypes. So we were fortunate, and I can explain how this works, but we were fortunate to identify an unrelated individual here at Texas Children's Hospitals with carrying the identical de novo missense variant heterozygous, sharing many phenotypes that we saw in the first patient. And we also, in another hospital, we identified a patient with that has the same origin in 163 mutated to not to a glutamate, but to leucine. So this also individual had similar features. So this was human genetics evidence supporting that this is likely to be a pathogenic variant. In our case, what we did was this gene called NOT is homologous to EBF3 as well as other EBF1, 2, 3, and 4 genes. And homozygous of this line or heterozygous, transheterozygous over a deficiency is lethal, which we were already knew from the literature. You can bring back the UAS fly knot gene and this becomes viable, showing that our system works. And also you can bring back the human EBF3 that causes, is viable, suggesting that the molecular function of the fly knot gene and EBF3 are conserved. However, when you reintroduce the R163Q variant or the L variant, these were lethal, suggesting that these were loss of function alleles. So this was one evidence. We have other experimental evidence to show that these are loss of function alleles using Drosophila. Another example is this case for WDR37. It really was a protein that we didn't really know what it does. It only had annotations as it has a WDR domain. We found 5 pediatric proband. We always start with 1, but if we find more, the patient has epilepsy, cerebellum morphology, coloboma eye defect, and developmental delay, and also had some similar dysmorphic features across different patients. And in this case, the gene was a CG. So when we knocked out the gene, we didn't know what to expect, but this was viable. So we're a little bit disappointed, but we try to figure out if there was any behavior defect. Because the patient had epilepsy, one of the phenotype that we wanted to look at was bang sensitivity, because this is kind of like the analogous seizure-like behavior. So you boltex the fly and look for the time for recovery. And indeed, loss of WDR37, loss of function mutation, had a bang sensitivity defect, which you can rescue by the reference WDR37. However, if you put in the point mutation that you see in the patient, you don't get a rescue. Again, this is showing a loss of function effect. Sometimes, we are not lucky that the human gene can rescue the flies. Well, if the variant is conserved in the fly protein, we can always play with the fly protein. So you can do, scarlet genome editing with CRISPR. You can recombineering of fly back construct, or you can play with the UAS fly cDNA to test the variant in the analogous fly protein to see if it also has a damaging effect. Also, this is something that also a lot of people that do in human tissue culture work, like using HEC293 cells, regardless of whether the cell actually has the endogenous protein or not, let's see if the overexpression of the human protein, wild type and variant, causes a phenotype and look for the difference between the two. And if you can see a difference, for example, if the wild type human protein doesn't cause a phenotype, but the variant does, that suggests that it's some kind of a gain of function protein. Maybe it could be a dominant negative, it could be a hypermorph, it could be a neomorph. Additional experiments needs to be done, but that's one way to actually do these studies. Or if maybe on the opposite, if the human protein just overexpression is toxic, you can actually see how that toxicity goes away or it gets strengthened. And this is based on just simple GAL4 UAS tests, and you can do this in different tissues. And I'll show you an example here. So this is also kind of one of the experiments where the humanization, actually we didn't attempt the humanization here because there were quite a bit of reagents that was already identified in developed in the fly field. So this was a case where a gene called RNF2, which is part of the polycomb complex, was identified with patients that had a de novo variant in this arginine 78 or S782R, a number of neurodevelopmental phenotypes. And just for your information, many of the patient that comes into the UDN have neurological presentation, maybe like I would say 70%. And in this case, the gene in flies is called sexcomb extra. SCO, loss of function, is lethal. There was a nice genomic rescue construct that was previously identified that would fully rescue this lethality. And because the two variants affected amino acid that were conserved, we actually showed that these variants were not loss of function allele. We actually also went one step ahead, and sexcomb extra is one of the polycomb complex that is regulating Hox gene through epigenetic mechanism. So we also wanted to see if the variant affects that specific function. We also, using a specific genetic allelic combination of sexcomb extra together with grainy head, we can actually make a fly that has this homoetic transformation in the leg, where you actually, the male flies have sexcombs in all 6 of these legs, and you can beautifully rescue that by the wild type copy. However, not the first one, the variant that's equivalent for the first variant. However, the second one partially rescued it, suggesting that this probably have a partial loss of function allele versus the first one is a more stronger loss of function allele. So you can also use it to compare the allelic strengths to correlate with the patient phenotype. And this is also another example that in this case, the patient phenotypes were immune deficiencies or cardiovascular skeletal abnormalities, which we didn't necessarily want to or thinking of modeling in flies. But because these patient actually had missense variant in the gene called TBX2, which is the homolog of fly bifid, which is a transcription factor that's required in a lot of tissues, including the eye, we did overexpression experiments. And if you overexpress the fly bifid, you can actually see that the eye gets smaller, and even the TBX2, the fly human homolog, actually makes the eye even smaller and associated with lethality. However, the two missense variants are partial loss of function alleles, shows a milder phenotype in both lethality and phenotype, and the eye morphology, and also we did electrophysiology on this, and that was also consistent. Once you do get engaged in this kind of a study, actually you get a surprise from, get a notice from OMIM and say, hey, we actually decided to name your gene. And one of the diseases, this RNF2, is now named Luo-Shock Yamamoto syndrome, having my name as the lost author of the paper and first co-author of the paper. They basically took these names, just like Alzheimer's disease was associated with, not Alzheimer's didn't have associated with Alzheimer, but the people who describe the disease often gets the I would say the honor of being part of a disease recognition. And there's another disease that I've also known. It's called a Ewan-Bellin syndrome, taking Wan Hee Yun and Hugo Bellin's name for this gene called OGDHL. So over the last decade, we've published 39 human disease-causing genes and 12 phenotypic expansions using Drosophila. And I think this is only just the tip of the iceberg, and we are continuing to work on this. We still have a lot of unpublished stories, 13 new genes and 22 phenotypic expansion, where we're actually trying to identify novel disease, more patients, or understand the disease mechanism. And also, some of these projects have identified novel disease mechanism that can elaborate into common diseases and druggable pathways that we're exploring. And we've recently published a review in Nature Review Genetics. So this is the link. If you don't have a subscription for this, just let me know and I'll be happy to send you the PDF. One thing that I want to post here is that functional studies using flies are inexpensive and scalable compared to vertebrates. I don't need to convince this audience, but we've been starting to do some calculations. So let's say that we get a gene to work on, and we have never worked on a gene. Well, we typically give it to a postdoc or student, and one postdoc can, let's say, handle 2 or 3 genes per year to characterize. A lot of the reagent generation, we have a system to generate these T2A Gal4, Kozak Gal4. A lot of the human cDNA is the reference. Even I told you that for 5,000 lines, there's already available things. So the only thing you need to meet the mutants. And making new reference lines is also not that difficult. And once we have these reagents, we need to characterize the loss of function phenotype. Characterizing the expression pattern is very important, and you can do this by the Gal4, the T2A Kozak Gal4 across the UAS-GFB, and that also helps us, doing some mechanistic study down the road. And then you can try to characterize the human protein based on rescue or overexpression studies. So typically, getting some idea about if this is a good project, it takes about 6 months to get preliminary data and year to solidify it. And we have, I think, our numbers is 60% success. So obviously, in order to publish this, we need additional 2 or 3 years to brush up the story, identify more patients, get into the mechanism. That adds the cost. But once you establish these assays, if somebody comes to you and they say, hey, I have another patient with a different variant, it's very easy to just actually do this for just one allele and then run the specific assay that we show that it works. So I think this is something to think about in terms of a clinical test. Maybe because there's so many people that have these VUS that needs to be developed, one application of Drosophila isn't really a clinical test. And obviously for genes that have a more high throughput cell-based assay, that's going to be much more cheaper, but always it's good to know what the functional consequence of the variant is in vivo, because you don't want to have some unanticipated results when you actually translate your finding to the patient. People kind of think I'm maybe crazy. It's like a fly test as a clinical test. Well, here's one example. So pregnancy test, this is something over the counter that you can do. You pour urine on it, and if somebody is pregnant, it's actually, you see a band right there. And that's because HCG, that's secreted from the human placenta, is a marker for pregnancy. And before this test was developed, people were doing what's called a frog test, where they injected a urine of a woman that wants to know if she was pregnant or not. And if the frogs start laying eggs, that's a sign of pregnancy, because the same HCG can work on the frog to actually stimulate ovulation. So we can maybe envision a time where maybe fly tests can be adopted into the clinics to classify VUS. So people that have these clinical exome sequencing VUS can take a fly test, and they would basically, based on that, they'll get a designation whether they're likely pathogenic or benign. I just wanted to say that the approach that we've been using for rare diseases can be applied to study the mechanism or study the functional consequence of variants associated with more common disorders. So the left is a published study from my lab and Mike's lab that we use this to study functional consequence of de novo missense variants that are found in patients with autism. And in this study, we actually tested the functional consequence of 79 variant missense variants. We found 30 of them to be disruptive, and we went on for one of them, a more deeper study, to find out that gain of function and loss of function variant causes different neurodevelopmental diseases in humans. And also more recently, again, when the COVID was hitting us, we were thinking about how can we actually also leverage Drosophila to study, COVID. And because we were now good at making a lot of these UAS lines, and COVID, SARS-CoV-2 only had 21 protein coding gene, we decided to generate UAS lines for all the COVID proteins and coded proteins. And then also couple that with US human cDNA lines where virologists identified direct protein interactors. There was about 300 human genes that were found to be physically interacting with the viral genes. And based on overexpression studies or co-overexpression studies, we were able to identify some of the targets of these proteins. So these are all published work. So if you're interested, I would recommend you to go to these papers. And I just want to spend the last maybe 5 minutes, if it's okay. I wanted to really re-emphasize that fly genetics, not only just us, but a lot of you can probably do the type of experiments. What I was telling you, just overexpression rescue-based assays are something that a lot of fly labs do in their labs. Maybe generation of reagents could be difficult, but we're actually happy to help you with any of these. But also, I think the really important thing right after the patient get a diagnosis for these rare disease condition is because they don't have a therapy, they want a therapy. So a lot of these patients start forming patient organizations, patient support groups. They do a lot of advocacy groups and also fundraising events to facilitate the research for their conditions. And once they actually raise the money, these patient organizations say, okay, we have all this money, who should we give it to? And we think that we should try to couple them to the scientists who already have expertise in the specific genes that they were actually interested in, because a lot of these cases are monogenic disorders, and model organism geneticists like fly geneticists, I mean, obviously cell biologists can also play a role using like CRISPR-based technology and high throughput screening stuff, and also molecular biology structure. Everybody has a role to play in here, and people have different expertise in different organ system. If you work on biology, we want to facilitate more collaboration with this. The problem is a lot of the scientists, maybe many of you here, are not part of a medical school. So how do we interact with these clinicians or patients who are interested in finding you? And also some funding bodies may have some grants money that they want to give out to people with specific proposal, but they can't really find people that are not, especially if you have some preliminary data that's very interesting, you haven't published, you're not visible. We developed this tool called Model Matcher, and this is also a website that modelmatcher.net. So you can actually enter your gene of interest and your expertise of interest. If you have a gene that you're actually interested, but you don't want to let the rest of the world know, we have a way of limiting the access to that information for certain people. But we were trying to collect information from scientists anywhere in the world. Which genes are you interested? Which model organism you work in? And that registry could be searched by clinicians. So in this case, I put in my gene called Notch1, and you can see my name, as well as there's an anonymous scientist, because that person said that I'm interested in Notch1, actually Notch1B in zebrafish, but I don't want to express who I am unless people tell me that they're not competitors for us. We are also partnering with the Canadian, European, and Australian initiative that's trying to make a registry of model organism scientists and get their profile of genes of interest. So if you go to Model Matcher, you can get all this information. And the clinician can actually look at your profile and say, hey, I want to contact that Shinya Yamamoto working in fruit flies on the gene Notch. They can just click my profile, send me an email. Or if they're actually really want to actually reach out to that zebrafish individual, then the clinician needs to, or the patient needs to provide their information, send you an email through the system, and you can decide whether to respond or not. So in summary, I think what we're trying to do through these database-like efforts, as well as maybe more advocacy work or raising awareness in collaboration with organizations or gatherings like Alliance of Genome Research, DIOP, Gene to Function, Monarch, RDMM. We really want to actually change how the world collaboration works. So the scientific research community is great and have a lot of knowledge, but many of them are not connected to the clinical research or the rare disease patient community. So what we want to really do is push you guys so that they're actually visible and they know about you. And also, we're also working closely with the clinical research community that is associated with different rare disease initiative, as well as patient organization who's really want to facilitate research so that we have this Venn diagram that has a larger overlap. I want individual of you guys to think about that. What you're doing in the laboratory, maybe your project is an Evo Devo project that has nothing to do with the patient, but if that gene is conserved in humans, there could be some experiments that you can do to help people's lives and maybe sometimes save them. And I want all of you to also think about when you're doing these things, there's a lot of resources, including things that are offered through the DGRC, Bloomington Stock Center, the TRIP, DRSC, as well as the stock center around the world, and also most importantly, Flybase that we actually get a lot of information. Pay your user fees, but I think all of them are trying to really help you, and hopefully the world would be a better place, understanding more curating human genes that are relevant to disease with the power of phylogenetics. Genetics. So with that, I need to thank a lot of people, but I didn't really want to put in a lot of names. So really thank Hugo Bellin, Mike Wangler, Oz Kanja from Baylor, Texas Children's. The Undiagnosed Disease Network is really a great collaborative environment with a lot of this is just a PI meeting, so you can think about how many clinicians or patients are behind this. Really want to thank the patients and family. These pictures are actually taken from a public site, so we have permission to show them, but these are beautiful kids that's trying to really live a life that's happy life with their families. And maybe other initiative, human genetics initiative that we're working with, funding agencies, philanthropy groups, as well as the resources that are actually really that's supported through the DGRC, Bloomington Stock Center, and other public stock centers. So thank you so much, and I'll be happy to take any questions.
Well, let's thank Shinya one more time, and we're happy to take questions and relate to Shinya as well, or you can just unmute yourself and go ahead.
Was that a lot of information? Okay, oh, I see something in the chat. Anna, thank you. Okay, so which method do you recommend for developing transgenic methods in Drosophila clone?
Isn't that cool?
Oh, hi, Claire. Yeah, so which method do you recommend for developing transgenic models of Drosophila? Cloning or CRISPR? So I think, so the word transgenic, actually, for me, we think that, you bring in a foreign transgene. So in our case, when we're actually generating things like UAS fly cDNAs, UAS human cDNAs, genomic rescue construct, we do cloning and then into a vector that has the APP site, and we put it into a docking site. So that's kind of an extra copy of the gene. When we want to actually engineer the fly genome in one way or the other, maybe make an analogous variant in the fly genome, I'll definitely use CRISPR. And also you can use CRISPR for other things. You can try to humanize the fly genes by inserting a human cDNA into the first exon, for example, and have a stop so that you're actually expressing the human gene at the more endogenous level. So one of the drawbacks, I would say, sometimes it's a benefit, sometimes it's a drawback, is that the humanization method that I talked about is still a bit of an overexpression. So because GAL4 UAS system tends to amplify anything that you're trying to express, even if you try to, re-express the human protein and where the fly protein, should have been expressed, because we're depending on the Kozak GAL4, the T2A GAL4 binding to the UAS human cDNA, we often get some overshoot. And sometimes that's actually deleterious. Sometimes that could be beneficial, because if the human protein doesn't have the 100% activity of the fly protein, but if you overexpress it, you might be able to get that. So it's one thing to think about, that humanization that in our strategy, sometimes it doesn't maybe work because it's not that the human protein doesn't work in the fly, which I think happens for 20% of the time. But I think in the 20% of the time, the other 20% of the time that's not working, it's just that you're actually expressing too much of the human protein is that's causing some damage. So I think both strategies, both the transgenic methods and the CRISPR-based methods, are very complementary. And there is some benefits of doing just overexpression assays as well, because sometimes if you knock out the fly gene, you don't really see an obvious phenotype, and that could be because there's other redundant genes or some kind of a regulatory mechanism. But then if you do an overexpression based strategy, overexpression of a protein give you some kind of a sufficient phenotype, and then you can see if that phenotype differs between the reference and the variant. So that's when the transgenic strategy becomes useful. So it all depends on what the property of the genes there are and proteins are that you're dealing with. And this is just a trial and error thing. I think there's no rule that tells us how much homology do you need for a human gene to rescue a fly gene. We don't have an answer to that, because sometimes it rescues with very low homologies, but sometimes it doesn't rescue with very high homology. Have another question, Scott. For genes without current T2A insertions, can we request that specific gene to be prioritized? And the answer is yes. So I don't know if actually you can see it on, so go to Hugo Ballen's website, and then there's actually a place where they say GDP, I think, and there is actually a place where you can find the currently existing T2A Gal4 and Kozak Gal4 lines. And some of them are in the pipeline, but even if they're not yet in Bloomington, you can find that if they're in the pipeline. If you don't see them, the best person to email is Oz Kanja and Hugo Ballen. Just email the two. I can put their email addresses down here, but if you say that who you are, and what genes are interested, I think they'll be happy to put it on their pipeline. Amit, great talk. Hi, Amit. Some of the fly genes do not have various isoforms like humans. What is the best strategy to address the phenotypes in human disease using humanization approaches? Okay, great. The word, maybe I should ask maybe clarify the word isoform. So when human geneticists talk about isoform, they typically think about splicing isoform. From the same gene, you're generating different splicing isoform, which also happens in flies, obviously, but human genes typically have more splicing isoforms. The other isoform that biochemists use is actually, they include the power log as also an isoform. So for example, the gene that I talked about, EBF3. So there's EBF3 is one of the 4 genes in this EBF family in humans, EBF1, 2, 3, and 4. And flies only have one, just a gene called NOT. And that's because in the vertebrate lineage, it underwent 2 rounds of whole genome duplication. And there's a good number of genes that were kept in 4 copies, like Notch is one of them, right? Notch 1, 2, 3, 4. And then EBF 1, 2, 3, and 4. All the delta, you have 1, 3, and 4 in humans. There's only 1 delta in flies. Jagged, there's 2. Serrate and jagged 1, 2. So sometimes the number of power logs differ based on the requirement. And for the EBF3, I showed you that you can actually rescue the lethality of the NOT mutation just by bringing in EBF3. And when we did the experiment with EBF1, it also rescues. And if you do this experiment with EBF2, it also does rescue. So what it tells you is that the function between EBF1, 2, and 3, and probably 4, is that they're molecularly the same. But in humans, once you actually underwent divergence, they started being expressed in different tissues, more or less, whereas the fly mutant is like a quadruple knockdown. So as long as you bring back the human genes in all the places where you need it in the flies, as long as the molecular function is conserved, you can do some experiments that's very meaningful. And so that's sometimes we do have experience for many of the genes that have these power logs, like VPS26A and B. You can also rescue the VPS26 single gene. But there are genes that we found that you can't do that. And one of the beautiful examples is mitofusine. Mitofusine, there's only 1 gene in flies, MFN, mitofusine, and the human have 1 and 2. We tried to rescue the mitofusine mutation with 1, it doesn't rescue. 2, it doesn't rescue. If you put both in, it rescues. So surprising thing about this is probably because the molecular function of mitofusine primarily is to mediate mitochondria-mitochondrial fusion, but it has a secondary role in mitochondrial ER tethering. And between the mitofusin 1 and 2, that function seems to have diverged, so that 1 actually has more mitofusin and 2 has the mito-ER fusion. So for survival, you actually need both activities. So this was a rare case where I think where you do need two of the human genes to put it back into the fly genes. But I think the reality is, the most cases, one can basically typically contains the molecular function. Yes, I hope that answered your question. Okay. And David Biller, thank you. Great talk, great. And Robin, what if a human cDNA can't rescue the loss of function of Kremik, Kozak, Gale-4 layer? What are the good alternatives? Yeah, so I think it depends on what your question is. So if the human cDNA can, so if the purpose is, again, to test what the functional consequence of that missense variant is, if an overexpression of a wild type protein has a phenotype and the variant doesn't, then you can say it's some kind of a loss of function. You need to be a little bit careful about interpreting overexpression because it's not physiological, right? But it at least tells you that the two proteins are different. And then if it's different, you can actually start asking the question, why is it different? Is the protein stability different? Is the protein localization different? So you can actually start, going towards that route as well. But if your purpose is to really try to model the condition in the fly and in a manner that's much more, clinically relevant, I would say, yeah, try to knock in, because the T2A Gal4 base rescue has its own caveat. Again, it's slightly an overexpression, so it might not work for a lot of places. So especially if you work on a variant that's conserved between flies and humans, definitely, yes, you have much more options, because you can try to knock in the analogous variant, right? The problem is that if you just focus on those, then sometimes very interesting variants that we get submission from the clinical sites, we can't work on, say, hey, well, this variant is not conserved in flies. We can't work on that. That's what the C. elegans people do, because their primarily method of doing functional study is the knock-in type approach, which actually works. And they do have a high success rate, because the chances are that if you have a really conserved amino acid that's mutated, the variant has functional consequence. But we actually are very also interesting in cases where, like, that variant is actually conserved over evolution in vertebrates. So all the vertebrates has it, invertebrate doesn't. So that still suggests it's probably important. And then the question becomes, can we study it in flies? So we do the attempt, and if the humanization works, yes, great. We have a great system to start working on these things. But again, with a little bit of a caveat, because sometimes we might not necessarily capture all the function of a human protein. So we go very strongly to the clinical sites if the results are positive, saying, hey, we have positive results. This looks like it's a damaging variant. We don't call it pathogenic yet at that point, but this is a damaging variant. However, if we get a negative results, right, we don't say that to the clinicians, hey, look, we think this is a benign variant, because we don't want to say that, because the fly experiments might not capturing some of the physical interactors that the human protein has. So negative results, I would be a little bit more cautious and say this was an inconclusive. And also similar thing, sometimes we knock out the fly gene, no observable phenotype. If I give it to maybe a behavior geneticist, maybe they find something that's really, really subtle, but we don't go that depth. And then when you overexpress the human protein, wild type or mutant, you don't see any phenotype. That's kind of a dead end as a project as well, because again, I wouldn't say it's actually positive or negative, right? It's inconclusive. And I would just go back to the clinicians and, hey, well, we did all these experiments. Here's the results. And, we don't think the flies would give you an answer. We should think about alternative methods. In that case, maybe we should go to, mouse models, zebrafish models, or even iPS cell-based models. Ali, yes, thank you. I hope that answered your question. Thank you, Shinya. Regarding the model matcher, is there a way to search for the human ortholog of the fly gene we are working on? Specifically, I'm interested in genes that might be submitted by physicians. Sometimes we are working on one gene in fly or two more genes. Oh, yeah. So the way model matcher works is that we ask people to put in their genes of interest based on their model organism and nomenclature of interest. So if you're a fly person, you can click on I work with flies and then type in your fly genes of interest. So you don't have to look up your human genes of interest. So when you do that, then in the back end, we'll basically convert that to the possible human ortholog. So if you put in, for example, Notch, I just searched Notch 1, but if you search Notch 2 or 3 or 4, my name would also pop up. And that's all based on the DIOPT filters, or actually the algorithm that Claire has been developing, and has been, so it's also very difficult. I mean, I would wish that AGR would at one point come up with a grand Excel file that tells you which genes are orthologous in all these species, but there's a lot of genes where the orthology relationship is a little bit complicated, right? Sometimes you have like 5 different fly genes and 3 different human genes, and they might be all orthologous to each other because at the last common ancestor, you only had 1 gene. And in the human lineage, it actually quadruplicated and maybe 1 had been lost. In the fly lineage, insect lineage, it might have been duplicated on its own because it was very beneficial for insects. So in that case, all 3 human genes are orthologous to 5 genes, and there's no one-to-one relationship. So that's actually a tricky situation. Which fly genes would you be studying in order to actually try to study, which of the human genes? So a lot of the cases, things are simple. If you only have 1 fly genes, 1 human gene, or maybe 1 fly gene, multiple human genes, things get a little bit more trickier when it's actually, 1 human gene correspond to multiple fly genes, or multiple human genes correspond to multiple human genes. And I think that's something that I think the model organism, or the database community, can actually take a look at to kind of give us a guidance of what genes could be more interesting to study. Yes, Philippe, thank you. And Amit, thank you. Author, last question from Ali, and we should wrap up the seminar. Okay, so yeah, so that was the last. Yes, we should wrap up.
Cool. Thank you very much, Shinya. It was really a whirlwind through a lot of different things. And thanks also for highlighting all the research centers and basically the nature of fly research, where, there's so much of coordination and collaboration between lots of groups, especially one specific thing about the Hugo Bellin group and the BCM group is that they donate all the reagents to DGRC and order it back when they need it. So thank you again.
It's actually cheaper than us keeping all the stock. So once we actually send it to Bloomington or DGRC, we actually, maybe not also always the cDNA, but a lot of things are coming directly from Sue's lab. But actually, yes, all the fly stocks, we trash it. And when we need it, we reorder back. So we do pay the fee to get it back. And but that's actually cheaper for us in the long run.
So yeah, thank you. And thank you all for joining us. And if you have any recommendation for future speakers, just email us at dgrc@iu.edu, or either Arthur and myself. And hope to see you soon in spring with another edition of the Lucy Cherbas Symposium.
Yes.
Thank you
Thank
all.
you again. Yep.
Thank you so
Thanks,
much.
everyone.